Hello, everyone. My name is Megan, and with me I have Edward, David, and Caroline, and we will be presenting our senior design project, which is called EcoFAB, Advanced Microbiome Science through Fabricated Ecosystems. So microbes are everywhere, and they play a prominent role in agriculture, soil composition, ecosystem function, as well as health, disease, and medicine. They are one of the most complex living organisms in the world. And if I were to take soil from my backyard and Caroline were to take soil from her backyard, we would have two completely different microbiomes. And this is really hard for microbial research because it means that these studies can't be reproducible. However, researchers have found 16 isolates that are found in abundant consistently in the rhizosphere of the soil. And the rhizosphere of the soil is where the roots are. And this means that wherever you take soil samples from, as long as it's where the roots are, you will find these 16 isolates, which means that we are able to have reproducible results. And this is exciting because now we can start understanding how these microorganisms act, as well as manipulate these microorganisms and push them to act a way that we want them to. However, we can't manipulate these organisms through their genes because they are able to have horizontal gene transfer and able to mutate. And due to this issue, we can't control the genome and have them act a certain way like we can, for example, when we genetically modify plants. However, what we can do is find metabolites that lead to interactions that we like through their metabolic capabilities. So in our project, we want to understand the microbes' metabolic capabilities of carbon. And in order to do this, we took 95 different carbon sources and fed 16 of our isolates. And I'll let Edward take it from here.
To execute the experiments, the procedures were divided into three different stages. Stage 1 consists of isolating single colonies by plating and culturing. First, 16 isolates from an open switch gas field in Oklahoma were isolated, and these isolates were shipped into the autoclave to observe the desirable outcome of isolates and rule out any potential contamination. From the culture plate, isolates were inoculated into the culture tube and grown in the tube until the distinctive growth was observed. Stage 2 consists of washing, pipetting, and normalizing to a fixed starting cell density. First, the isolates were washed by multiple centrifugation to separate isolates from the media. Then these isolates were pipetted into biolog plates with 95 different carbon sources to characterize the favorable carbon sources. These were prepared in triplicates to eliminate any potential human errors. Also, concentration of isolates were normalized through dilution of media. Then isolates were measured through the spectrophotometer for initial measurements and measured again after 1 week of growth. Stage 3 consists of analysis, visualization, and interpretation. We used Python to generate the heat map. Using the data from the heat map, isolates were classified based on their metabolic intake of the carbon sources using dendrogram. Then principal component analysis was performed to verify the results of dendrogram. Let's go into the results.
So the heat map that was generated from our data shows how each of the carbon sources affects the growth of the microbes. It could be seen at the bottom of figure 2. Each row represents a different carbon source from the 96-well plate, and each column represents one of the 16 microbes. The dark regions represent the wells that had the highest level of growth. Now, in general, growth shows that the organism has the means to metabolize and use the carbon source we provided. And the purpose of this heat map is to quantify which carbon source can be metabolized in each isolate. And when we do that, we can begin to look for similarities amongst isolates. We are able to group the 16 microbes into 3 categories based on their metabolic capabilities by looking for trends and patterns. And then they were confirmed by using the software we wrote that takes in the raw data and generates this dendrogram seen on the top of figure 2. We can see that there are clear similarities in which carbon sources are preferred amongst isolates in the red group, which varies from those generally preferred by the blue group isolates, as well as those in the yellow group. So for example, we can clearly see that the isolates in the yellow group are very different to each other, are very similar to each other, because for the most part, they did not seem to metabolize any of the 95 carbon sources at all, as also seen in the heat map. Principal component analysis, or PCA, as seen in figure 3, is another way we can visualize the previously mentioned results. The blue, yellow, and red points correspond with the blue, yellow, and red from the dendrogram and the heat map. The general clustering of the isolates in the same group can be seen, further confirming that there are clear metabolic similarities in each of the 3 bacterial groups.
So with the clusters we obtained from our results, we can now look into the interactions with microbes of the same cluster. We could also look into other factors that affect growth, such as testing phosphorus and nitrogen sources. Once more data is gathered regarding these sources, our next step will be to build synthetic communities of microbes that could be used as an alternative to fertilizer. Ideally, these communities will provide similar growth factors and be resistant to abiotic stressors. To do this, we will test the isolates in an EcoFAB to study the effects that they have on specific crops or plants. The EcoFAB consists of a growth chamber and allows for the study of host organisms in gnotobiotic conditions. We will feed the rhizosphere through the medium and then identify the beneficial bacterial microbes to design further synthetic communities. Other studies will be done looking into these communities to help revitalize soil that has been depleted of nutrients due to overcropping. We would like to thank Dr. Wheeler for guiding us through the project, as well as Deepon and Dr. Karsten Zengler for mentoring us throughout the experiment and study.